This experimental approach combines BrdU incorporation with fluorescent in situ hybridization to quantitatively analyze the replication timing of mammalian chromosomes. First, incorporate BrdU into live cells and harvest the metaphase chromosomes. Then perform DNA FISH combined with sequential BrdU antibody staining to identify the individual replicating chromosomes. Next, use photomicroscopy to capture images of FISH signals and BrdU incorporation in individual mitotic figures. Using cytovision software, calculate the area and intensity of the pixels represented by the BrdU or DAPI signals on each isolated chromosome. Results can quantify and compare the BrdU incorporation in individual rearranged chromosomes and their homologs.
Because the method described here evaluates individual cells, it has the ability to detect changes in replication timing on chromosomal rearrangements that are present in only a fraction of the cells in a population. Compared to the recently developed high-throughput microarray and DNA sequencing protocols, this method allows for a direct comparison of homologous chromosomes residing within the same cell. It also has the ability to identify unambiguously chromosome rearrangements that affect chromosome replication timing of entire chromosomes. Demonstrating the procedure will be Leslie Smith, a research associate in my laboratory.
Seed the cells at approximately 70% confluence.